I'd like to start by expressing my gratitude to the members of the Academic Symposium Steering Committee who have worked really hard to make this event a reality. Their commitment to showcasing a lot of the college's original research, scholarship, and creative achievements of our students, of our faculty, of our staff is so appreciated. This is a great day in the year. Last year was my first academic symposium. You might say a little bit of my introduction to McDaniel, and it's one of my favorite events. So I would like to actually recognize by name those individuals on the steering committee, and if they happen to be here, to stand so that we can collectively thank and recognize you. Dr. Wendy Morris, back there. Wendy chaired the steering committee. Dr. Alexander Shampoo Crawley, also Ms. Michelle Gribben, Dr. Michael Pollan, Dr. Gerard Sivion, Dr. Maureen Williams, Ms. Yang Hong Wang, Ms. Jessica Victoria, Ms. Cheryl Nauer, Ms. Valerie Royer, and Ms. Mary Jo Colbert. Thank you so much. It's now my pleasure to introduce our keynote speaker, Dr. Allison Kerwin, Assistant Professor of Biology here at McDaniel College. Dr. Kerwin is a marine symbiosis ecologist who investigates microbial, ecological questions using biostatistics and experimental techniques, with a particular focus on host microbe and microbe-microbe interactions in marine symbioses. Her research aims to understand the effects of environmental changes on these symbiotic systems. Dr. Kerwin received her BA in biology from Smith College. She went on to earn an MS in marine biology from Northeastern University and completed her PhD in microbiology from the University of Connecticut, where her dissertation focused on the stability, development, and function of symbiotic bacterial communities associated with the Hawaiian bobtail squid. Since joining the biology department at McDaniel, Dr. Kerwin has taught a range of courses, including principles of biology, microbiology, invertebrate zoology, and marine biology, among many others. She has an extensive publication record in top-tier journals with several works currently in preparation. She's frequently invited to present her research at national and international conferences. And today, Dr. Kerwin will deliver her keynote address titled Under the Sea, the Impact of Insecticides on Jellyfish and Corals. However, before I invite Dr. Kerwin to the podium, I have the privilege of adding yet another honor to her list of accomplishments. I'm pleased to announce that Dr. Kerwin has been selected by her faculty peers as McDaniel College's Charles A. Belkey Jr. Engaged Faculty Fellow. She didn't know that was going to happen. This award was established with a gift from Charles A. Belkey Jr., who's an alumnus of the college and a member of the McDaniel Board of Trustees. This award recognizes Dr. Kerwin's exceptional dedication to engaging students in original research and experiential learning opportunities. Since arriving at McDaniel just 3.5 years ago, Dr. Kerwin has mentored 5 students in completing independent research projects that led to their biology capstone. Dr. Kerwin's mentorship enabled students like Devin Maranto, Jose Pack, and Michaela Patterson to persevere in bioinformatics research projects despite interruptions that occurred during COVID-19. Jose's work was even accepted for a talk at a national conference. Dr. Kerwin has remained a supportive mentor as these alumni have gone on to graduate school and medical school programs. More recently, Dr. Kerwin guided students Juliet Beer and Dev Goodman in hands-on research characterizing metabolic properties of jellyfish bacteria, imparting specialized lab techniques to those students. She has also implemented an innovative course-based research experience called Tiny Earth in her symbiosis class, training students to discover novel antibiotic-producing bacteria. Perhaps most exciting, Dr. Kerwin was awarded a prestigious $500,000 grant from the National Science Foundation to study the effects of insecticides on jellyfish development. I suppose we'll hear some of that today. A project that will also fund summer research opportunities for 12 McDaniel students over the next 3 years. Dr. Kerwin, would you mind coming to the podium? I'd like to present this certificate, which is something to remember the Belkey Award by, and also invite you to take the microphone. I look forward to hearing your talk.
Thank you so much. Wow. Well, thank you so much for that wonderful introduction, Provost Lilly. I appreciate it. And thank you to the organizing committee for inviting me to talk today. So I am going to be giving you some of the background that led me to the proposal that Provost Lilly mentioned that was funded by NSF, and talk a little bit about the goals of that project.
Will you please talk into the mic?
Is that better? Okay, thank you. So I want to start by just giving a little bit of background on what a coral is. So here we have a individual coral polyp, and it's made up of clear tissue, which you can see here at the top. And then there are these small photosynthetic microbes that live in the tissue that are sort of a greenish brown color, and that's what give corals their colors. Each coral head that you see on a reef is made up of hundreds of thousands of these individual polyps that are all working together in a colony. And then you get hundreds and hundreds of corals forming reefs together. Some of these reefs get so large that you can actually see them from outer space, and so this is an image of the Great Barrier Reef around Australia from the International Space Station. Corals are really, really important economically to people, and so in the United States, the coral reef fishery is worth about $100 million per year, and then in addition to that, you have tourism activities, so people go to reefs for snorkeling, for scuba diving, but then they also utilize local restaurants and businesses and hotels, and that industry in the United States alone is estimated to be in the billions of dollars per year. In addition to those sort of straightforward economic benefits, coral reefs also provide a lot of really important protective services. So when waves are coming into shore, and you can see here in blue, they spend a lot of their energy on healthy coral reefs. And so by the time they actually reach the coastline, the wave energy is drastically reduced, and the wave height is quite a bit reduced as well. And if the reef is instead degraded, which is in orange here, the waves don't have any way to spend that energy, and when they hit the coastline, they're still pretty substantial. And so in this study, they actually see that the whole reef, on average, spends about 100% of the wave energy that hits it. There was also a study done during Hurricane Ian down in the Keys where they were predicting pretty severe storm surge coming in. But as the hurricane passed over this restored reef in that area, the wave height was reduced by 90%, and the storm surge was substantially less than predicted as a result. And so these reefs can really be incredibly important to protect human life and property on the coastlines as well. Now, in addition to those economic factors, reefs are these incredible hotspots of biodiversity. So if you think about the ocean environment, pretty large area, coral reefs only make up about 1% of that area, but 25% of marine species are found on reefs. So they really just have this incredible diversity of organisms there, and we can equate them to sort of a tropical rainforest in a terrestrial setting. So in addition to having all of these diverse corals, and you can see some fish in these pictures, you'll also see things like giant clams right there. And then this little guy, this is a nudibranch, or a sea slug. And a fun story that some of my students have probably already heard. So when I was doing my master's, we were in French Polynesia, and we were out on this dive. And I happened across this little friend. And I was so excited because I've always loved nudibranchs, and I just couldn't believe I was getting to see one in the wild. And so I was really engrossed, and I took this picture and really spent some time looking at it. And then we get back on the boat, and everybody's chatting, and they're all talking about this sea turtle that they saw during the dive. I had seen no such turtle. I was so interested in this nudibranch that the turtle just passed right on by me, and I had no idea it had been there. So you can see where my focus is on dives. So another thing that coral reefs are really important for is new sources of drug discovery. So one stat that I saw while I was putting this talk together is that 25% of new anti-cancer drugs are coming from coral reef environments. And so you can see some of these examples here of compounds that have come to fight cancer. And this is one example of a coral that has produced an anti-cancer drug. And you might be thinking, well, why would corals be so good at producing anti-cancer drugs? And one theory is that because these corals are sessile, they can't move around, they're stationary, they have to fight for space for other organisms that are growing near them. And so they do that by secreting compounds that actually attack rapidly dividing cells. And so they can, maintain their little area on the reef. Well, cancer is also a disease of rapidly dividing cells, and so we can take these compounds used for competitive purposes by these corals and apply it to human medicine. Another example of a drug found on a reef is from these cone snails right here. So these are extremely venomous organisms. You don't really want to meet one in the wild if you can avoid it, but they have been the source of a new painkiller. And so if somebody is going through chronic pain or a really severe pain episode, then you will have this, and opiates are no longer working for them effectively. You can use this painkiller from the cone snail, and it will actually still be effective, even when the opiates no longer are. This sponge here in orange is has produced a pretty cool compound. So some of you may have heard about the antibiotic resistance crisis currently facing us, where a lot of bacteria are resistant to the drugs that we use to treat them. And so they have found this compound from this sponge that actually reverts bacteria to a susceptible state, even if they have developed resistance. And so that's something that's still under investigation. And then the last one I'll mention here, this is a sea whip, and it was found to contain this really mild anti-inflammatory. And it's been a really nice addition to a lot of skin creams and things like that to reduce redness in the face and possibly, maybe treat a little bit of eczema or psoriasis as well. So lots of really important benefits from coral reefs, right? But unfortunately, reefs are really threatened right now. So 75% of coral reefs worldwide are either threatened or endangered. One of the factors affecting reefs is overfishing. And so 55% of reefs worldwide are experiencing overfishing right now. As we have depleted some of the fish stocks on reefs, technology has gotten better, and so we're able to pull in more and more fish. This is a snapper haul off of a coral reef. And so sometimes we take too many fish, and the reef actually, that population of fish can't replenish itself because there's been too many of them taken. Another really troubling, destructive fishing process is this use of dynamite fishing, where they will actually throw dynamite in the water and explode it, and it stuns all of the fish in the area. They float up to the surface, and then you can just easily take them out of the water. But as you might imagine, it also has some pretty devastating effects on the reefs themselves, and pretty much demolishes those so that you no longer have that habitat, and it's going to be very hard for the reef to come back at that point. So Sort of on the other side of things, you also have this issue of invasive species. And the example I'll use here is lionfish. So lionfish are native to the Pacific Ocean and were not originally found in the Atlantic or the Caribbean. But in 1985, we had our first sighting of a lionfish in the Caribbean. It's thought that probably somebody had bought it at a pet store and had it in a tank and then released it into the ocean when it got too big. And over the next 15 years, they started to see occasional sightings here and there. But around 2000, 2001, this population really took off. And these fish are kind of amazing. They can produce 31,000 eggs every 1 to 4 days. And they reach sexual maturity in less than a year. They also have no real predators in the Caribbean except for groupers, which are unfortunately overfished. So not much out there to take care of them. And they prey on the juveniles of a lot of our commercially important native fish. So they've had a really destructive impact on these reefs. And during this period in the early 2000s, as the lionfish population sort of went through this boom, there was a 65% reduction in native fish species. And you can see by 2013, all of these red dots are lionfish sightings. They go all the way up the East Coast as far as Rhode Island. So they've really done an incredible job of invading this ecosystem. Pathogens are also a big problem for reefs. And so here I'm going to focus on an emerging pathogen that's been found down in the Florida Reef Tract. It's called stony coral tissue loss disease, or skittled. Kind of a funny name for a pretty serious problem. So here is an example of a coral where you're seeing just a couple of patches of this disease pop up. This is the beginning of February in 2019. And over the next month and a half, the entire coral dies. So it's a really fast-acting disease, and it also unfortunately infects a lot of different species. So most diseases are only going to attack a specific species of coral. This one is pretty indiscriminate and will take out a lot of different types. And so it can have really devastating impacts on reefs where it's found. So first discovered in 2014, it had never been seen before in this one little section. So this is the tip of Florida here down into the Keys. And the red area is where you see the disease. The green is where it hasn't been reported yet. And over the next several years, skittled spreads more and more. By 2018, it's reached all the way north in the Florida Reef Tract. And then it continues, and by 2021, has taken over the entire reef tract, including down into the Dry Tortugas National Park. And it's unfortunately also now made it out of Florida and into the Caribbean, and all of these red dots are places where skittled had been reported as of a couple of years ago. So it's really become a big problem in the Caribbean and is just devastating reefs all over the place. One other place that we're seeing some impacts on reefs is from terrestrial runoff. So this is when you have, for instance, a river system and there is agricultural land on it, or there is maybe some sewage runoff or development going on. And so all of that ends up in the river, which then leads out to the ocean and drains there. And so if you look at what you should see near a river mouth, there's going to be an area generally where there's not going to be a lot of coral because there's too much fresh water coming out from the river. But as you get further away, there should be a return of the coral cover. But when you add this runoff and this higher nutrient load, the corals have a lot of trouble with that. They like very nutrient poor environments with very clear waters. So this is at a river leading out into the Great Barrier Reef area, and 20 kilometers away, so about 12 miles, you have this area that is completely dominated by macroalgae, so red and green macroalgae here. 40 kilometers away, so maybe 25 miles, you still have a macroalgae dominated ecosystem, so the corals are still not able to live there. By the time you get to 60 kilometers and then finally 70, you start to see hard corals, and then you get into a more healthy reef ecosystem. And so that can be a really big problem, and it takes out large swaths of reef that would otherwise be able to live there. Okay, so then the really big one, right, is climate change. We are seeing the climate shift worldwide, and that is having a big impact on our reefs. So This is a graph showing the temperature around the Great Barrier Reef. This is the average temperature here, and then the blue would show that there is a lower temperature anomaly. The red is that there is a higher temperature anomaly. Corals can survive a lot better if there's lower temperatures than higher temperatures. They don't do well with those high temperature stressors. And this is starting in 1860. You can see that we really don't have very many high temperature anomalies until you get to maybe around the mid-1900s, and they've steadily been increasing ever since. And so we're having more and more of these high temperature events in the waters. In addition to that, we are putting more and more carbon dioxide out into the atmosphere, and that is starting to absorb into the ocean, where it kind of messes with the buffering system for pH in the ocean. And so over time, that can actually start to reduce the pH through this process of ocean acidification. And so this blue line here is showing you that reduction in pH. And I will say, we're not seeing a big reduction in pH. If you look at this scale over here, it's just dropping a little bit so far. But pH is really not supposed to drop in the oceans at all. It's supposed to be pretty stable. And so even these initial indications are troubling. So why are these a problem for corals? Well, I mentioned at the very beginning that corals have these photosynthetic microbes in their tissues. Depending on if you've ever learned about these, you may have heard a different word for them at different times. So zooxanthellae or zogs. Symbiodinium was for a while the genus that they were all assigned to, and then that was expanded to a family called Symbiodiniaceae. I know those are all mouthfuls, so I'm going to call them photosymbionts for the rest of this talk. So these are photosynthetic symbionts. And so this is that same picture. You can see them in this coral tissue here. This is what they would look like under the microscope. And so when a coral is happy, these photosymbionts are living all throughout their tissues. That's what gives the coral their color again. But if you go through a thermal stress event or some other stressor, the coral will actually expel those photosymbionts from its tissues. And so at that point, the clear tissue is all that's left, and so you see straight through that to the coral calcium carbonate skeleton. The coral can still recover at this point, so the coral, if the stressor dissipates, they can regain those photosymbionts and become healthy again. But if it goes on for too long or if it's too severe, then the coral doesn't make it and it gets taken over by macroalgae. And so here's an example of that. You have a healthy reef here. Then a couple months later, it's gone through a thermal stress event, and so you have this bleached, stark white coral. It unfortunately was not able to recover, and so then the reef is become covered with macroalgae, and that structure will begin to break down. Now, I'm going to switch gears just a little bit, but we're sticking with climate change. So one other big factor with climate change that may at first not seem super related is that insect population ranges have shifted as well. And so I'm going to focus here on the mosquito Aedes aegypti. This is an important mosquito vector for a lot of different human diseases, things like Zika, dengue, yellow fever. And these mosquitoes prefer really warm environments and more urban environments where there might be some left out small freshwater cups and stuff like that that they can breed in. And so they are really only going to be spreading disease when they are happy and there is sort of this nice warm temperature that they can be active in. And so in these maps, we're seeing the months per year that transmission is possible for this species. So the red color would mean it's active all year round. The blue, it's only active every once in a while. And so in 2019, we can see Florida right here. It's sort of in the middle of that range. And if we look at this scenario of unmitigated climate change, where we really don't do much to stop it, by 2050, Florida has definitely moved up that scale, and by 2080, it's pretty much up here in this solid red range. And that's a problem because we're also seeing an increase in certain diseases down there. So dengue is something that's very troubling disease. You don't want to get it. And there's always going to be cases of transmission from people that have left the country and come back in from international travel. But at least as far back as 1935, there were no local transmission cases of dengue in Florida. And so in 2009, we suddenly see this outbreak of dengue that lasts from sort of 2009 to 2010, where we start to see local transmission from this expanding mosquito population. That dies down a little. There's another small outbreak in 2013, and that is actually a different strain of dengue. So there's not just one strain circulating in Florida, there are multiple strains. There are a couple of cases in this range that you can't really see because of the scale of the graph, but then we have another decent outbreak in 2020, and in 2023 there are about 170 cases, I believe, in Florida in that year of dengue. So a big problem, and one that the Florida government is obviously interested in trying to curtail. And so back in 1949, they create the Florida Keys Mosquito Control District, or FKMCD, which is a government entity with the sole mission of controlling mosquito populations. And so in 1949, they're mostly looking at nuisance mosquitoes. They're going around, they're like, okay, there's sort of more mosquitoes here and there, and they're using DDT. And so this is this incredibly effective insecticide that can really take out all different kinds of insects, and it is marketed as being extremely safe. And so some of you may know that that's not exactly accurate. So in 1964, this book, Silent Spring by Rachel Carson, comes out, and she is able to put these pieces together and show that DDT is actually not that safe. So what's going on is that as insects consume DDT, other animals eat the insects, and it works its way up the food chain in sort of this process of what we call biomagnification until it gets to large birds of prey. And we're starting to see these big decreases at this time of these birds of prey while DDT is being used. And what is happening is that the DDT is actually interfering with calcium deposition in their eggs, so that when the bird goes to lay on its eggs and incubate them, the eggs are not strong enough to withstand that bird's weight, and so they get crushed. There's also some human health implications. So it's thought that possibly the use of DDT is responsible for a decrease in sperm count and an increase in endometriosis during the 1900s, and there's still work being done on that. So DDT is banned in the US in 1972, and the Florida Keys Mosquito Control District switches insecticides. So for the next 30 years, they're really just looking at nuisance populations again. If there is a sudden influx of mosquitoes in an area, they go and they treat them. But by 2000, we're really needing some broader control mechanisms for the reasons I've already put out there. So they start these spraying campaigns, and they're using aerial spraying. So for adult mosquitoes, they use this insecticide called Nailid, which they spray from helicopters and planes. And so this is still going on till this day. So I went online over the weekend, and this is the spraying that happened yesterday on Sunday. And so this is Key Largo right here, and this bright green area is the region that they were spraying with this insecticide yesterday. They also do larval spraying. They actually use a bacterium for this. It's something some of you may have in your houses to treat houseplants for fungus gnats, or to actually just get rid of some mosquitoes on your properties. And so they spray that as well from helicopters and planes. And then if you get a higher concentration of mosquitoes popping up somewhere, they do truck-based spraying with two other insecticides, permethrin and malathion, where they just go to those areas and spray as needed. So how do these insecticides work? So we've got these three insecticides that I mentioned, Nailid, malathion, and permethrin. Nailid actually degrades into this other compound called dichlorvos, which is also of concern, so I'm including it here. And so Nailid and malathion are organophosphates, whereas permethrin is a pyrethroid, which is a little less toxic to people. But all three of them are going to work against the nervous system. So they are going to bind to GABA receptors in the insect, and those are neurotransmitters, and so they basically shut down the nervous system and kill the insects that way. Nailid and dichlorvos have a secondary mechanism of action where they actually will work against, or they will work to increase reactive oxygen species. So these are toxic oxygen molecules that can cause a lot of problems in these insect cells. Now, all of these are very effective against insects, but they can also be toxic to aquatic invertebrates. So at certain levels, they can really cause a lot of problems. And so then the question is, well, what levels are they at, right? And so when you're doing aerial spraying, there's going to be some of these insecticides that end up in the water. So in this study, they were going out after some of these aerial spraying campaigns or truck-based spraying campaigns. So this is about permethrin, which is from the trucks. They were looking at the wind direction, and so that's that yellow arrow there. They will actually only do the aerial and truck-based spraying if winds are under about 10 knots because they don't want them going where they don't need them. But what they found, and this is in Key Largo again, so that same area that I showed you they were spraying yesterday, is that downwind of the spraying, they were detecting permethrin in the water in the Florida Bay. And so these orange triangles are somewhere between 1 and 10 micrograms per meter squared. And then this one site here with this red circle is 10 to 20. So a decent amount is ending up in the water. They did this again on a second date, and they got very similar results. So again, we're not really seeing it in the Atlantic at this point. And then they did it a third time, and the wind was in a very different direction at this point. This was actually right before Hurricane George went through the Keys. And so at this point, with the wind going towards the south, you've got insecticides that ended up in the Atlantic as well as in the Florida Bay. And they, for the first time, test in these canals that are inside the key, and they get this very high concentration of, I think it was 52 micrograms per meter squared in that one site. And so that's also probably accounting for some of these higher levels here as things get washed out of these canals into the Atlantic. Now, this study was focused on permethrin, so the EPA estimates that a safe level for inverts is about 0.01 micrograms per liter. This is different units because they used filters over here, but I think you can tell these are a lot bigger than that one, right? So not looking great. And then they also did a little bit of work looking for Nailed, which is the aerial spraying adulticide. And so in this first survey, they did find Nailed in the Florida Bay and at pretty low concentrations in the Atlantic. And the second one, they found some in the Bay as well. The third one, because of that hurricane going through, they had actually evacuated the helicopters and airplanes in advance of the hurricane, and so there was no spraying going on, and they didn't test for Nailed in that case. I'll also mention there was this one other study where they were going to do some lab tests of insecticides, and they were bringing in seawater from the Keys to work as their negative control. So it was supposed to have no insecticides in it. And when they tested that water, they found that it had consistent levels of about 0.5 micrograms per liter of permethrin and 0.7 micrograms per liter of Nailed in just the regular water that they were pulling out of the ocean. And so they weren't able to have a negative control for the study because there were these baseline concentrations persisting in the ocean. Okay, so can these pesticides impact cnidarians? Well, wait, what is a cnidarian? That's a fun word there, maybe people don't know. So cnidaria are this phylum that includes things like jellyfish and corals, as well as other things like sea anemones. And so if you think about what a phylum is, for us, we are in the chordate phylum, so everything that has a backbone, basically, would be our relatives. These guys are sort of cousins to each other when you look at corals and jellyfish. So, pesticide exposure can indeed impact corals. And so there hasn't been a ton of research done on this yet, but the research that has been done is focused on the larvae, which is pictured right here. This is an example. So in this coral right here, Acropora millipora, when there is low levels of permethrin in the water, it decreases larval settlement. So these larvae have to go find an area to attach to the surface, and then they go through metamorphosis, which was also decreased, to become that coral polyp that I showed you in that other image. For Porites asteroides over here, they looked at low concentrations of nailid, and they found that that decreased larval survival as in that species. So we do have some indications that even low levels of these insecticides can have troubling impacts. And so all of that has led me to this project that I am currently working on, where I look at how insecticides can impact the upside-down jellyfish that Provost Lilly mentioned has been funded by the NSF, the National Science Foundation. And so just a little bit of information on what an upside-down jellyfish is. So I think most of us, when we picture jellyfish, you're thinking of something that's sort of going through the water like this, right? Tentacles streaming behind it. These guys are a little different. So when they reach adulthood, they flip over and they go down to the bottom and they just lay on the bottom, on the sandy bottom of the ocean, their tentacles up. And so this is one picture of one taken from a lab I was in at Penn State, and then this image is actually showing some on the seafloor, and I've gone ahead and outlined those in yellow so you can see where the jellyfish are in that image. So they then just stay they don't really move around very much. They pump water over themselves to catch food, and they also actually have photosynthetic microbes in them, those photosymbionts that allow them to use energy from the sunlight. Okay, so I spent an awful lot of time telling you about corals. Why am I studying jellyfish? Well, first of all, they're much easier to maintain than corals are. So if you want to maintain corals in a laboratory setting, you've got to get just the perfect amount of light. Your water has to be absolutely perfect. There are just a lot of really finicky conditions that you have to get perfect. Jellyfish are a lot easier. This is my tank setup in my lab right now. It's just some regular old aquarium tanks with some filters in them and an air stone, and the jellyfish are just fine in there. The other really nice thing about these jellyfish is that they have this prolonged polyp stage, which you can see here. And these polyps will actually go through this process where they create buds that can turn into new polyps. So you can see one right here coming off of this guy, and then there's one up here. And so they will swim off on their own, they will attach to the surface, and they will grow into a new polyp. And so you can go from having just a few polyps to having quite a lot of them in a fairly short amount of time. And that's actually what I've been spending most of this semester doing, is trying to get a lot of polyps growing in my lab to be ready for summer research in a couple of weeks. Another really nice thing about these jellyfish is that, unlike corals, they're really not considered to be a threatened species. So you've all heard at this point about all of the threats for corals, but the jellyfish are pretty abundant. And so this is one of the sites that I go to collect jellyfish, and it's just, there's like a low bridge right here, and I'm looking down at that. And there are actually hundreds of jellyfish in this photo. And so I know they're a little hard to see, so I've gone ahead and outlined a bunch of them in yellow for you. All of those are individual jellyfish right there. And that's just sort of in the foreground of the picture where there's not too much glare, right? So that is really helpful, because if we were going to go try to get corals to do this research, I've told you how decimated the Florida reef tract is. It's really hard to find healthy corals at this point, and if you do, it's questionable if you should take them, because maybe they should stay on that reef and try to, help rebuild the reef. In addition, you'll see that there are these mangrove trees in the background here, and so these jellyfish polyps actually grow on decaying mangrove leaves that drop down under the trees, and so they are found a lot among mangroves. Mangroves are one of the primary sites where we have these spraying campaigns, because mosquitoes also love mangroves. So that makes a really nice connection there as well. Now, in addition to all of that, these Cassiopeia, these upside-down jellyfish, are also really good model organisms for tropical corals. So they have these same photosymbionts in them that the tropical corals do throughout their tissues. And so they're able to use this energy from sunlight to get nutrients from these photosynthetic microbes, and then they can give waste products back to them, and you have this nice even exchange. In addition to that, they have a very similar bacterial community to what is found in corals, and so that's work that I've been doing over the last several years, where we look at what bacteria are found in wild jellyfish, and it's very similar. So that's nice. And so having that similar system means that we can really use these as a model, and when you figure out things about these jellyfish, you can apply it to corals as well. So this is my first project goal and the one we're going to be focusing on this summer. We are going to be looking at how the presence of these insecticides at different concentrations impact the polyp stage of the life cycle of these jellyfish and also the metamorphosis. And so this is when the polyps actually get colonized by those photosymbionts and go through this process where they change into a swimming medusa. And I have 4 awesome students working with me this summer, so Patty, Gwyn, Mackenzie, and Angelin. I think all of you are at this table right here. Yay! So they are going to be working with me on this project this summer. The other big part of this goal, oops, that came up early, is to look at how the insecticides impact these interactions between the bacteria and the photosymbionts and the jellyfish host. And so this is building on some work that I've been doing here over the last couple of years. And so I had 2 students, Dev and Juliet, working with me a couple summers ago, and we had a bunch of jellyfish bacterial isolates that they went through and categorized all of these different metabolic properties. And so we can now have that as sort of our default, and we can say, okay, if we add insecticides into the equation, how does that change things? I'm also really interested in looking at how the bacterial groups change over time, and so this is some other work that I've been working on where I've seen that this one group, Nitrospiraceae of bacteria, changes during that developmental metamorphosis process. And what we think is going on is that when those photosymbionts come into the jellyfish, they actually change the environment inside the jellyfish host, and so the bacteria have to adjust to that. And so some of the genes that seem to be implicated are those involved in nitrogen metabolism and denitrification, and so it seems like these bacteria could be involved in nutrient cycling in the jellyfish, and we're really curious about whether adding insecticides in might change those nutrient cycling processes. So to end here, I want to just talk a little bit about how I got here. So I started off doing some undergraduate research myself, and so I really wanted to do a year abroad when I was in college. And so I went to Geneva, Switzerland for my junior year, but I wanted to do research. And so I looked around while I was over there, and I found somebody at the Geneva Botanical Garden who was willing to take me on as a student. And I spent the year studying moss development, of all things, right? I then took what I think sometimes is now called a gap year. It was quite a few years, where I went and worked in a bunch of different labs. And so I did some work on lizard malaria and scorpion genetics. That was at the Museum of Natural History up in New York. And then I worked on fruit tree viruses with USDA down in Beltsville. And so from there, I went and I was getting my master's degree. And there was, early on in that process, a class we were in. It was on professional development, and everybody had to stand up in the front of the room and say what they were interested in, what they wanted to do later on in life. And so I get up there and I say, I want to study the anthropogenic effects on corals. And the room is like, what are you talking about? What do you mean that's what you want to study? And so that means that I wanted to look at how we as human beings are impacting these coral reef ecosystems. And it's taken me a while, but here I am now, right? And I am looking at these insecticide impacts on coral reefs. So I didn't, it took me a little bit longer to get there. I really had no background in microbiology at that point at all, but I sort of got into that right afterwards, studying this temperate coral. I then worked on squid, as we heard about during my PhD. And then I got into this jellyfish world during my postdoc at Penn State, which I have brought now here to McDaniel and have now got this funded grant. So with that, I'd like to thank my lab, so all of the undergrads that I've worked with since I've been here. I'll also thank the Medina lab. So Monica Medina was my postdoc advisor and is still supported. She is the source of the jellyfish that are in my lab right now. This is the Key Largo Marine Research Laboratory, which is run by Bill Fitt. And so this is the view out the back door. You can just walk out and right into the water, and there's jellyfish right there that you can pick up and bring inside. And then finally, just the McDaniel community. So the friends that I've made since I've been here, my colleagues, and especially my students, I wouldn't be able to do this project without you. So thank you so much, and I'm happy to take any questions. So I think there's one right here.
How do you make the connection from working in a microenvironment to a macro environment?
So the question was, how do I make this connection from working in a microenvironment to a macro environment? And it's an interesting switch. So you really have to think about all of these different ways that you're controlling the system so that you can make sure that the micro results that are difficult to see can still be investigated when you're in more of a macro setting, like for instance down at this research lab, and you have to keep things sterile and stuff like that. But looking sort of at how these microbes work with these more macro animals is just fascinating, because pretty much every organism out there has microbes that are playing some crucial functional role. We have all kinds of gut bacteria, as well as bacteria in our hair and our skin that are playing really crucial roles for our health. And every animal that we investigate, we see similar things. And so sort of figuring out how they work together, I think, is a really important part of biology.
Thank you. That was wonderful.
Oh, thanks.
I'm curious. You shared the concentration of some of the insecticides in the Florida Bay.
Mm-hmm.
Do you have plans in your lab to demonstrate like dose response relationships?
So we are planning to look at different concentrations and to see sort of what that cutoff is that is actually considered harmful, because I do imagine that there are low enough concentrations where I think any of these inverts would probably be fine, and so figuring out where that cutoff is, I think, is really important. Yeah.
So you mentioned that one of the places you're sourcing the jellyfish are from those mangrove areas where there's the insecticides and everything, and I wonder if you've done any work on figuring out how much the concentrations are in the tissue since they're staying in the waters, and how you would get kind of a baseline of like how much is in the tissues of those jellyfish before you work on them this summer, or if you have like a lab population.
So the question was, do I have a way to figure out sort of what the insecticide concentrations are in the jellyfish tissues if they're coming from these areas where you're having insecticides treatments applied? And so I am not actually using those for this study. I am using a lab population of jellyfish that's been established for quite a long period of time, both through Penn State and then through other labs as well. And so at least the expectation is that they've been in the lab long enough that there should not be any insecticides in there, because we keep, starting new populations of these polyps. So it shouldn't be a concern. Famous last words, but Yes?
Do we understand the mechanism by which the photosymbionts are ejected from the coral? And is that a process that could potentially be inhibited to prevent that coral loss?
So I think we sort of understand it, I guess, is my answer to that. There are a few different hypotheses out there about how this is working, and there's been a lot of arguments over the years about exactly what's happening. Oh, sorry, the question was, do we understand the mechanism by which the photosymbionts are being expelled by corals during bleaching events? So there has been some work looking at that and trying to figure out how to trigger them taking the photosymbionts back up more quickly. It's difficult to prevent them from expelling them altogether. So they actually live in the tissue that's right along the stomach of the corals, and so when they don't want them anymore, they can kind of just attack those cells and they make it a really hostile environment. There's some research that suggests they may actually consume some of the photosymbionts, and so preventing that from happening would be really tricky. Yes?
Do you know if there's any potential for a controlled plant of native species that is a natural mosquito repellent that can be done?
I mean, so the question is there any plans to try to put in some native species that might repel mosquitoes? I think that would be really tough. It's a pretty big area. I mean, I love the idea, but I think we need to figure out some new methods for controlling mosquitoes, certainly, and so adding plants that might have some of that repellent is important. But also there is a lot of education underway, especially in the Keys and other places in Florida, about making sure that there is no fresh water left anywhere outside of houses, so that you can reduce places where they can breed, and really trying to lower some of that down. But it's going to be impossible to get rid of that, I think, altogether using methods in that way. But you can try. Yes?
Any questions, but based on your studies in the future, do you think you will be able to draw a conclusion or recommendation which direction the pesticides should go?
I mean, that's certainly my hope. So the question is, should I be able to draw a conclusion about these insecticides and their effects by the end of my research? And that's definitely my goal, is to be able to say, okay, this specific insecticide seems to be really causing a lot of problems developmentally for these jellyfish, which means that it's probably also doing the same for corals, and maybe we need to reinvestigate what concentrations are safe to apply, and find other insecticides that are better. And so one thing, even since I submitted this grant application originally, that they've done is they've started spraying for the larvae using this bacterial solution. And those bacteria are pretty benign and are fairly safe for most other organisms. And so it might be a really nice thing if we switch more and more towards that and away from the adulticide spraying. And if we control the larvae, that might be a more effective use of spraying campaigns. So I think, having that data out there that you can apply can then help inform these conservation decisions.
I'm interested in understanding the grant.
Mm-hmm.
Do you feel like $500,000 seems like a lot, but is that how far will that get you, and what does that really look like when you're using it for your students and yourself?
That's a great question. Okay, so the question was, how far does the $500,000 get us? So that's funding for 3 years. It is allowing me to get a lot of equipment that I need to do some of this research. So, for instance, I have this really cool incubator coming in the next couple of days, actually, I think, where you can control the light cycle and the temperature simultaneously, and so you can really have the jellyfish in just these ideal conditions with the sufficient amount of light during these studies. And it lets me also do a lot of sequencing work. And so it should be pretty solid funding for 3 years. And this is actually a really cool grant program. So it's a new program through NSF that is aimed specifically at new faculty at small colleges. And so I was like, yes, that sounds great. And so one of the goals of this program, though, is to get people into a position where they can then go and apply for additional grants moving forward. And they have at least one conference mentoring development session scheduled for the second year of the grant, where they're going to bring a bunch of the recipients together so that we can learn more about that process and think about how to get more grants in the future to continue the work that we're doing. So to help the corals and the sort of broader, so the question is, what can we do to help right now? I mean, so a lot of it is, all right, I guess I would say take your pick. There's a lot of things you can do. So some of the stuff I mentioned, like overfishing, so making sure that you're eating sustainably caught seafood is a really good way that individuals can help make a difference there. also trying to get things like lionfish on the menu at different restaurants. So there's been a big push for that. They're supposedly very tasty, this like nice buttery white fish. There's been a big push to try to get those to be this commercially important fish, and they have all these derbies where people go out and catch as many lionfish as they possibly can, so that they can sell them. Looking at government policies such as how to control runoff and how to maybe mitigate some of these climate change effects is also going to be very important. So, it's one of those things that we really need to look at systemic change and pressuring elected representatives and government officials to make policies that reflect that, I think, is going to be really important. Oh, yes.
You shared a infographic from the Australian government with temperature anomalies since the 1800s. Was that global, local, and if it was local, how was that data collected?
Okay, so the question was how was the temperature anomaly data collected, and what was sort of the scope of that data? So that particular data set was local to Australia. It was just one of the clearer graphs that I found to use for a presentation. We're seeing similar data sort of worldwide. I don't know if I know for that particular study how they collected the data. I'd have to look that up, but I'd be happy to get back to you if you want to give me your information. Okay, well, thank you so much.